When we started talking about immunology, talked about four very important features. We have covered three of those very important features. The fourth one, specificity. I said that when a particular pathogen enters the body, our body only makes antibodies against that particular pathogen. So how does our immune system know which B cells to activate so that the B cells make antibodies against that particular pathogen and that particular pathogen only? So let's see how that works. So our helper T cells, they get, as we have already talked about, get activated by mature dendritic cells. Mature dendritic cells display B7, which is the second signal. This is the first signal. Again, we have talked about that. In addition to these two signals, when dendritic cells engulf pathogens that require antibody production, they make interleukin 4 and they use this molecule, the signaling molecule, to guide the naive T cell, helper T cell, into effector Th2, helper T cell type 2 lineage. So this again, mature dendritic cell engulfed a particular pathogen. Let's say this was a salmonella, and this little red ball is actually a peptide fragment from salmonella. So after this T cell has been activated, it will encounter B cells. Now we have in our lymphoid organs, there are a lot of different B cells displaying many different antibodies. We have talked about antibody diversity. The B cells will also, those B cells which are displaying an IgM molecule attached to their surface, which is acting as a B cell receptor or BCR, if this IgM molecule can bind that same pathogen, in our case it is salmonella, this binding of IgM and the pathogen partially activates the B cell, causing internalization or engulfing. The B cell will engulf this pathogen. When B cell engulfs this pathogen, it will degrade it and it will also display fragments of this pathogen, salmonella pathogen, on its surface. I have said that B cells also possess MHC2 molecules. They are also antigen presenting cells. So they will display the fragments of our salmonella pathogen on their surface with the help of MHC2 molecule. This, if those B cells that can display the same fragment which was displayed by this antigen, our mature dendritic cell, when these fragments, the same fragment is displayed on the surface of these B cells with the help of MHC2 molecule, remember our helper T cell recognized this particular structure on the surface of dendritic cell. It is going to recognize, when our helper T cells recognize the same structure on the surface of B cell, they activate that particular B cell. The B cell, after it has engulfed that pathogen, salmonella, it will become mature B cell. By becoming mature B cell, I'm implying that it will start making another protein or surface protein called CD40. CD40 happens to have a counterpart on the surface of helper T cells. When helper T cells become mature, they start making CD40 ligand. So now this B cell is going to get two signals. The first signal is coming by when the B cell engulfs or phagocytoses the pathogen, salmonella in this case. That's the first signal. The second signal is coming from CD40 and CD40 ligand. Now our B cell has received these two signals, and this B cell now can become an effector B cell, which is in our case a plasma B cell. This B cell will now produce antibodies. This will, when it becomes mature, remember the B cells start producing IgM antibodies initially, which will no longer be embedded in the plasma membrane. They will be released in the extracellular space with the objective that these antibodies can now go and find the pathogen and bind it. What happens after antibodies bind the pathogen? We'll look at that a little later. But here, this process basically ensures that our immune system is going to have a specific response against a particular pathogen. So this was basically the B cell activation by the dendritic cells. I would like to tell you that initially when we talked about the reoccurrence of a particular infection, we said secondary response is much greater. One of the reasons why it is greater is, remember we said that B cells, when they become mature, they also divide. Some of the B cells, when they get activated, they divide. Some form effector plasma cells, the other cells become memory cells. Now, if we have abundance of these particular memory B cells that can recognize Salmonella, which was the pathogen in this example we talked about, we have number one, higher number of B cells. Number two, these B cells will use a shortcut. They will bind the Salmonella pathogen and they will act as antigen presenting cells. So it is a shortcut, and that is why secondary immune response is much more rapid and much more robust.